Okay, so hello, good afternoon, everyone. Welcome to the live interaction session for week 12. This is officially the last interaction session for this course, for the 12-week course. This is the week 12 session. And following this, we have another interaction session, which will be an extra session for discussing some of the topics that are yet to be covered that we could not do before because of lack of time. And apart from that, we'll also be going over the entire course. We'll just quickly brush over all of the topics that we have covered as part of the entire immunology course. If there are any weak points that need to be discussed, we can take those up. If there is any query that anybody has, we'll address those. And of course, we'll also try to figure out what are the weak points that need to be focused on more and whatever changes are required for the same, and overall help you in addressing the questions that are more relevant to the examination that you have ahead and for any examination that you might have long term. So with this idea, we will begin today's discussion. Now, as you already know, the topic for this week was centered entirely around vaccines, vaccinization, immunization, and of course the idea of um, and of course, the idea of generating monoclonal antibodies also, which are being implemented as vaccines in today's time. So I have not covered monoclonal antibodies today because I think most of it was discussed quite extensively by Dr. Shradeep, by Professor Shradeep. And we can go over the hybridoma technology maybe in the extra session if time permits, or if that is a query point for anybody. But apart from that, we'll mainly be focusing on vaccines. I will be focusing on some details about the topic that has already been covered. And of course, we'll be going over the previous week's questions to address any queries that might lie there. And apart from that, we will also be trying to look at some examples apart from the ones that have already been cited by Professor Shudi for the types of vaccines that are commercially being used in today's time, and how they have been relevant, what are their advantages, disadvantages, etc. So that's the whole idea that is going to be covered for today. Now, let us begin with today's session. So for week 11, the topic centered around week 11 was mainly tolerance and autoimmunity and the advantages and disadvantages of transplantation and how that how that has been changed over time and how that has been made more commercially viable over time. Uh, but for now, let us just go over the questions quickly. I might be toggling a little bit between previous week's questions and this week's just to help it out, but let's just quickly go over the concepts. So the first question was about the term xenoimmunity, which refers to A, the immune response to foreign antigen, the immune response to self-antigen, immune response to transplanted organs, or immune response to commensal microbiota. Now, the very term xeno by itself is reflective of something that is not a self component, something which is derived from something which is okay. Let's go with the terminologies again. If you remember, there was autologous. There's something which is an individual's own body tissue or own body derived substance that is being introduced into their own self, then that is autologous in nature. But something can also be derived from a different individual of the same species. But when the derivation, when the tissue or the organ that is being transplanted or that is being, let's not even talk about organ, let's talk about just a protein by itself. When it is being derived from an entirely different species, then in that case, it refers to xenoimmunity. So the immune response that gets induced in response to a protein or a, let's talk about organ, for example, when that is being introduced into my system or any other individual system who is obviously not of the same species, then it refers to xenoimmunity. So among the options that have been given, if we have to talk about a different species, then the correct answer in that case would be microbiota. So that would be because they are not of the same species and the immune response that is generated against them is xenoimmunity, xenoimmune in nature. Other examples you could maybe take, for example, the immune response that sometimes gets elicited in response to some valves which are taken from other organs. When I say valves, mainly when people have heart blockage, because their blood supply gets interrupted due to the, the buildup of fats and buildup of not just fat but cholesterol rather, along the chambers of the heart or through the blood vessels, often known as atherosclerosis. So in those cases, what happens is that over time, heart function or the ability of the heart to pump out blood effectively throughout the whole system gets compromised. And when that persists over a period of time, it can be extremely fatal. So in that case, people undergo different types of surgery depending upon how extensive that blockage is. And one of the preliminary technologies that used to be implemented was in, implantation of pig or bovine sources of valves, which were installed into these people, into these individuals, so that it could help their heart function work a lot better. Now, over time, there have been a lot of modifications over this to minimize the amount of immune response that comes up against it, to make the acceptance of the graft a lot easier, to make it more quickly functional without much risk, so that immunosuppression does not have to be extended over a period of time. So all of these modifications have been made over time to make it a lot more efficient. So that refers to xenoimmunity. Coming to the second terminology, just a second. Coming to the second question, the concept of autoimmunity was first described by Paul Ehrlich, Edward Jenner, Karl Landsteiner, or Eli Mechnikov. In this case, it is Paul Ehrlich, the correct answer in this case. Which of these is not an autoimmune disease? The options here are type 1 diabetes, rheumatoid arthritis, AIDS, and Graves' disease. Now, this is a question that can potentially lead to some confusion. If you have gone through the previous week's lectures, or if you've gone through the live interaction session video, you would know that all these examples, option A, B, and D, are examples of autoimmune diseases. We have discussed them in different contexts, how the body's response to some self-antigens changes because of XYZ reasons, and that leads to the autoimmune condition that we are talking about. But among these, while AIDS may seem like a very obvious option for an autoimmune disease, you must keep in mind that it is not autoimmune in nature. What it actually is an infection that, not infection, is a condition or a set of conditions often, which sets upon because of an infection by a particular virus. In this case, that virus is HIV, human immunodeficiency virus. What does it do? It leads to a systemic suppression of the immune response that an individual has. So as a result, the person themselves does not suffer from a disease, but rather their ability to respond to a disease, or their ability to have to elicit an immune response when a sort of pathogen comes in, that is extremely low. As a result, patients that often have AIDS, or for that matter even SCID, which is severe combined immunodeficiency syndrome, they have very, very minimal ability to respond to those infections, which any other normal individual would be able to sustain very easily. So what happens in those cases is that patients who have AIDS or who have SCID in that case, they often, when they suffer from something as minimal as, say, not minimal, but something as commonly encountered, like say a common cold or a diarrhea or a cholera, for example. Yes, cholera is a serious disease, but I'm saying that any other individual would be potentially be able to counter the side effects of that disease and would be able to sustain it. But such an immunocompromised individual is not able to do so, and therefore such secondary infections are actually what become very lethal for such an individual. So they have to live in an extremely careful manner with a lot of precautions so that they do not encounter diseases very easily, especially for SCID. That's a huge problem. So that was option 3. In this case, the correct answer is option 3, which is AIDS. Which of the following is a non-organ specific immune disease? So you might be familiar with most of the other options, maybe not psoriasis, but you can go through it. Psoriasis is mainly associated with the skin. It's actually very common. You might, have even come across somebody who has the disease, and it's not very obviously apparent to people. But yeah, a lot of people suffer from psoriasis, which is a very patchy, dry skin that gets inflamed very easily. And it is also it's prevalent. You have already come across Crohn's disease and multiple sclerosis in the previous class. And of course, the most obvious answer is that by the name itself, you can tell that SLE or systemic lupus erythematosus is a systemic condition. It's not constricted to any one organ that, okay, the brain or the heart or the liver or any one organ is getting affected. No, it's a systemic condition and it is known as lupus or SLE, more elaborately known as SLE. So that is the correct answer to this question. Then comes the question about the principal mechanisms of peripheral tolerance. What does it exactly include? So we had discussed central and peripheral tolerance in the previous class. So what are the mechanisms that actually are included under it? The options here are suppression by Treg cells, energy, activation induced cell death, or AICD, or all of the above. So you might already be familiar with it, but let's just quickly go back to peripheral tolerance. Just give me a second. I think it should be here. This is central. Yeah. So for peripheral tolerance, you can see these are the different types of options, that there is cell death, or there is AICD or regulation, or there is cellular energy, depending upon the type of antigen that is there, so as to prolong the duration that tolerance can be maintained. So via Treg cells also, this is being kept in tolerance is being maintained within the system so that you can sustain it for a longer period of time. And here we had discussed AICD and how apoptotic cell death or AICD has a role to play in both central and peripheral tolerance. So again, coming back here. So since all three options are relevant here, the correct answer in this case would be all of the above. Then coming to the next set of questions, which of the following options is the mechanism for induction of immune tolerance? Central energy, peripheral energy, clonal energy, or all of the above? Of course, the correct answer in this case is also all of the above. For immune tolerance, you need energy at all three levels, and therefore all of the above is the correct answer. The next question was about the function of the autoimmune regulator or AIRE gene. I had not elaborated on it very extensively, but if you've gone through the lectures, it was mentioned there. So you would remember how it acted as a transcription factor and what was its job basically. So its job was to promote the expression of tissue-specific antigens for T cells so that self-distinction can be done very properly, and autoimmune response can therefore be kept in check under normal conditions. So the correct answer in this case, of course, would be option C. So the other options are not true, which is inhibition of T cell development. No. Proliferation of T cells is helped. No. The correct option would be option C. Then, after exiting the thymus, mature T cells are subjected to the secondary selection, where majority of self-reactive T cells are deleted or are rendered anergic. So what is this process known as? So what you need to keep in mind is the location at which energy is being mentioned, because cellular energy is maintained at both levels. But we are specifically being asked about the energy that sets in after T cells have exited the thymus, and their selection is being carried out, and therefore this process would be known as peripheral energy or peripheral selection of T cells. So the correct option would be option B. The autoimmune thyroid disorder with a presence of anti-TSH receptor antibody is TSH is thyroid stimulating hormone or TSH receptor antibody is suggestive of Hashimoto's thyroiditis, Graves' disease, Crohn's disease, or psoriasis. So again, you would probably remember it, but let's quickly go over to that slide. I think it should be there. This was Hashimoto's, yeah. Here we had seen how anti-TSH antibodies or autoantibodies to the thyroid stimulating hormone receptor was leading to unregulated activation of the thyroid gland even without the correct stimulus by the hormone. So the gland is constantly being stimulated, as a result of which there is an overproduction of thyroid hormones and therefore Graves' disease, becomes an autoimmune condition in such people. So this is what we had discussed, and that is, of course, the correct option in this case. Just to quickly make the distinction between the two. Hashimoto's thyroiditis is a different condition. Your antibodies are being formed against thyroid proteins like thyroglobulins and thyroid peroxidase, etc. And these are leading to the infiltration by all these lymphocytes, or basically by the immune system, into the thyroid gland. And you can see that by the H&E stained images as well. So that's about going back to the questions. Yeah, so that was the correct answer. That is Graves' disease, option B. Finally, hemolytic anemia is caused due to the destruction of which of the following cells? The RBCs, WBCs, macrophages, or platelets. So hemolytic anemia, as the name indicates, even if you're not already familiar with it, is because of the destruction of RBCs or red blood cells, and therefore it sets in this autoimmune disease condition. So that is option A. So with this, we come to an end for the week 11 assignment. I think it should have been quite easily apparent to anybody. But if there is any doubt, you can certainly go ahead and ask. Somebody will help you out and will try to answer your queries regarding that assignment. Now coming on to the discussion topics for this week. So first of all, we talk about vaccines, and we discuss something you're already familiar with, which is the discovery of vaccines. So we probably had discussed this sometime very early on during this course, week 1 or week 2, somewhere around that time. So the earliest vaccination studies were done by these two scientists, Edward Jenner and Louis Pasteur. Particularly the English physician Edward Jenner, he made a huge advancements in the deliberate development of immunity. He had immense contributions, often also recognized as the father of vaccines or the father of immunity. So what he did was he reasoned that inoculating the fluid from a cowpox patient, which is a disease which was often contracted by milkmaids, that's how its discovery was actually done. So from a cowpox pustule, what is a pustule? It's basically what you see in this image. You can see that on the surface of the skin, you can see these sort of lesions which are present, which are extremely painful. You might have seen the sort of lesions which more commonly most of us actually come across it, which is in case of chickenpox, that people who suffer from chickenpox get all these huge pustules throughout their body. It's very painful, and of course, it leaves a mark, and it lasts for a sustained duration, like around 7 days or so, and therefore, in comparison, these pox, basically the pox virus has this, or the viridae, this entire family of viruses, they actually have this tendency to produce these pustules over the body of the individual who suffers from them. So from a patient who was suffering from cowpox, from their pustule, when the fluid that is present within that pustule was injected into other people, it was actually protecting them from smallpox, which is a related disease. So, okay, first of all, the idea is a little unusual, that why would you do that, but yes, it is serendipitous discoveries like this, or rather maybe he had some rationale at that point of time. He might have been studying immunity for a particular duration of time to have enough confidence over this idea, but that's what he did. And that's what led to the discovery of vaccination, that you can have prophylactic actions of certain infections if they are already present at very low doses within an individual's body, and that has a protective sort of role to play in such an individual. So what he actually did to test this idea was that he inoculated an 8-year-old child with this fluid from a cowpox pustule, and then intentionally infected such a child with smallpox, and it turned out that the child does not develop the disease. What you have for reference at the bottom is the type of lesion that you get in cowpox patients caused by vaccinia virus versus lesions of smallpox, which is caused by variola major and variola minor viruses. You can see that they are quite distinct. But yeah, that's what happens there. And therefore, this idea itself was revolutionary. Adding to these unbelievable discoveries by Edward Jenner, of course, such experiments are near about impossible to do today without proper ethics approval, because of course back then, this is the 1800s. So at that point of time, the rules and regulations were not as stringent. There was probably no governing body, no one was really answerable, and these patients, at that point of time, what mattered was that lives were being saved. So a lot of things were excused. In today's times, you need to get ethics approval, ethics clearance for everything. You must have the paperwork approved. Only then can you even consider implementing any as something as small as even the smallest drug that might be placebo in nature, which means that it doesn't actually have the drug, but is a inactive version of the same drug, or might just be saline in that case. So even for that, you need to have proper setup and proper approval from governing bodies, and that is something that are set in to be more mindful of the consequences of the actions of scientists and doctors in today's time. However, let's go back to the 1800s. And what Louis Pasteur did in 1885 was that he administered his first vaccine to a young boy named Joseph Meister, who we certainly have to credit for the same. He administered this vaccine, and then that child was bitten repeatedly by a rabid dog. Again, as you can see, there was no regulation back then, which is why people could do all this. And he was inoculated with a series of attenuated rabies virus preparations, and turns out that the boy lived. So there was protection against rabies as well. So this is actually a wooden engraving of Louis Pasteur, who was watching Joseph Meister receive the rabies vaccine. And over time, once this concept was developed further with more rules and regulations, with more scientists bringing in their contributions towards the field of immunization and vaccination, many diseases like diphtheria, common measles known as rubella, German measles known as rubella, mumps, pertussis or whooping cough, poliomyelitis and tetanus has declined dramatically as the vaccination protocol has become a lot more common. I will be discussing the vaccination schedule that is now almost compulsory for every child after they're born, depending on what age they're in. And these have become critical so as to conserve the not just that individual's health, but the country's health and society's health at large. So that's the overall goal. And what you see on the right are the different types of symptomatic manifestations of these disease. So this is common measles or rubella, mumps, diphtheria, and this is of course for whooping cough. I did not label it, but yes, that's what you see as the manifestation of these diseases that have declined dramatically over time. Here you can see a comparison sort of all the different types of diseases that have very significantly affected society at large. So barring all other, barring all other causes of death, you can see, of course, that cardiovascular conditions, they predominate very largely, and you have neoplastic diseases which come a very close second, which is cancer. Basically, you have injuries, asthma, perinatal conditions, etc., which contribute very, very, very hugely to mortality. But infectious diseases are a huge, huge contributor towards the deaths that occur across the world. Now, this is slightly old data because, of course, it's from an older version of the book. You have more updated records for the same. But the number of diseases, number of infectious diseases that are associated with mortalities across the globe is immense, mainly because most countries do not have the setup that is required for proper immunization. And in case a person suffers from disease, the follow-up, or not even the follow-up, the correct timely diagnosis and the correct treatment, the ability to afford such treatments is a huge problem for, especially for third world countries like India. Among the infectious diseases, you can see that annual deaths are credited majorly to respiratory infections, but we also have diarrhea and associated diseases, HIV/AIDS, tuberculosis, malaria, vaccine preventable childhood diseases still being such a huge contributor, majorly because a lot of people do not really get their children vaccinated right after they're born, or do not go ahead with the follow-up vaccinations or the booster doses for those diseases, which is why they sometime in their later life they do suffer from the disease. We also have meningitis, hepatitis B and C, tropical parasitic diseases, STDs, dengue, and leprosy being the largest contributors towards these diseases. So, having discussed the problem, let's now talk about solution. What is the solution? It is immunization. The process of immunization is eliciting a long-lived state of protective immunity. Otherwise, you can also call it as prophylactic immunity against a disease-causing pathogen. Exposure to the live pathogen followed by recovery is one route towards immunization. So you can have it's an accidental sort of prophylaxis whereby a particular individual encounters a disease and then again encounters the same pathogen at some point later in their lives, and therefore they have immunization towards that, or they have a protective immunity against that pathogen. The other possibility is using vaccines. So vaccination is the process whereby intentional exposure is made intentionally to forms of a pathogen that do not cause disease. You do not want the pathogenic version of the pathogen to be present because, of course, that won't be helpful. That might instead become lethal. So you go into some non-pathogenic or non-disease causing forms of the same pathogen to which you carry out intentional exposure. Now, there are two methods via which immunization can be done. A, it can be active. When we say active, it means that one individual who is getting immunized is actively responding to that pathogen which has come in, or either it is a attenuated or it's a kill pathogen, whatever it might be. And therefore, antibodies against that pathogen concerned is raised in an individual's own body. However, when antibodies are not raised in the person's own body, but instead it is raised in the body of other animals like other humans, horses, mouse, rabbit, guinea pigs, etc. When that happens, that is passive immunization, which means that the system or the host within which antibodies are being raised is actually what determines what is the type of immunization, whether it is active or it is passive. If raised in an individual's own system, then it is active. If raised in other individuals or not just other individuals, but other species, other animals, and then passively introduced into a host system, then that is passive immunization. Now, what are the different types of vaccines? This is all from Immunology. So somebody was asking in the YouTube lectures where these videos have also been uploaded. So again, I'm reiterating this, not just for copyright issues, but also because this is extensively been what I have followed, and I highly, highly recommend that book. However, for this particular chapter, which is on vaccines, you can refer to the other books suggested by Professor Shruti and Professor Ganguly as well. That might be more helpful for you because Kubi has very limited data and information on vaccines. So majorly you can refer to my slides. I have included some slides from my own study days in college. So that material is also there with credit to my professor. And apart from that, we also have a lot of information from Kubi. Some basic information from Kubi will always be there in my slides. But yes, please refer to whatever is most comfortable for you to understand the concept. And of course, internet is at your fingertips, so you can Google anything and everything, go to official websites and study from WHO, study from the Indian Association for Pediatrics for what is done in India. And, you can be constantly updated regarding the same. So when we talk about human vaccination, these are different classifications that are made for human vaccines. The first set is whole organism vaccine, among which we have live attenuated, inactivated, or killed. Inactivated or killed vaccines. So among these, we have a set of live attenuated vaccines, which are present for diseases like measles, mumps, polio, which is the Sabin vaccine, rotavirus, rubella, tuberculosis, varicella, yellow fever, etc. What is the advantage? It gives you a very strong immune response. Often even comes along with lifelong immunity with very few doses. Problem is that it requires refrigeration, its storage, and its circulation to different parts of different countries is actually difficult. And thirdly, the fact that it is attenuated, but obviously is not killed, allows it or gives it that scope to mutate again to the virulent form and can become disease causing again in over the course of time. So that always remains a risk and a disadvantage when you talk about live attenuated vaccines. The second set is for inactivated or killed vaccines. So we have cholera, influenza, hepatitis A, plague, polio Salk vaccine, and rabies. So you can see that there are two types of vaccines for polio. One is the Sabin vaccine, one is the Salk. The Salk vaccine has an inactivated or the killed pathogen, whereas the Sabin vaccine has the live attenuated pathogen. So this is a key distinction between both of these terms. Please be attentive when you, in case you get a question regarding this. It was not directly mentioned in the lecture, so it might not be very relevant to you with respect to this course. But for your own knowledge, for your own information, be very mindful of it. Now, when we talk about inactivated or killed vaccines, they are a lot more stable. They are safer than live vaccines. It has a lot less chance of actually mutating and going back to the virulent condition. Its refrigeration is not very necessary. It's a lot more stable at room temperature, and therefore its shipping is also a lot more hassle-free. The only disadvantage being with it is that it's a weaker it induces a much weaker immune response in comparison to live vaccines. And more often than not, it requires a booster shot to be accompanying it. Booster basically means that you are boosting the immunity against that particular pathogen by repeated inoculations with the same pathogen. So it actually accesses the memory of the immune system, and therefore it elicits an overall or an altogether stronger response in the long term. Next, coming to the second subtype, which is for purified macromolecules. So among purified macromolecules, we have different subtypes. You have already come across these in the video lectures by Professor Shrudeep. The first type is a toxoid. Now, what is a toxoid? It is an inactivated exotoxin. Okay, let's understand the terminologies one by one. Toxoid is different from a toxin. Why so? A toxin is something which is getting directly synthesized and released by the pathogen, and therefore it is very, very pathogenic in nature and is capable of causing disease. Injecting that will certainly not be helpful, and therefore you need to somehow modify it or alter it so that its immune potential is being or its ability to induce an immune response is being conserved without actually causing the disease. So that is for that condition to actually be possible, what is done is that an inactivated exotoxin is synthesized. Why are we saying exotoxin again? Because this type of macromolecule is synthesized or is after synthesis is being secreted into the surrounding environment by the pathogens, which are diphtheria and tetanus in this case, and then it is being inactivated and is being used for patients. I will briefly be discussing this eventually over the course of our discussion today, so you can refer it to there. Its main advantage is that the immune system becomes very well primed to recognize such bacterial toxins and therefore can counter these eventually in the future. Let's take one example. Say, for example, you might have often heard that when you get a deep cut from a very rusty sort of metallic source, say scissors or some nail, you prick yourself somewhere, and certainly if that is something which has breached your skin, the epidermis basically, you're always suggested to take a tetanus vaccine. The idea behind that is usually these rusty areas, they obviously any area has any bacteria, for example, but these regions, say for example, if you have a scissor or you have some sort of a furniture in your home which is metallic in nature, which has rust gathered on top of it, more often than not, it has a very high possibility that such a region is probably rich in some sort of bacteria, particularly in Clostridium tetani, which is the bacteria that causes that secretes the tetanus toxin, and therefore it's very dangerous. Particularly why it's more dangerous is because this bacteria, as you can tell, Clostridium tetani, it's anaerobic in nature. So what it does is it doesn't grow on the skin surface. It grows very deep within. And what it effectively does is that the tetanus toxin will end up causing, it will not allow the muscles to function. It will compromise muscles function and will completely freeze them. So therefore, this particular vaccine, you will find it very commonly in any pharmacy. For example, if you eventually encounter a cut, you can take a tetanus vaccine and the shots can last you like 6 months to a year, depending upon what is the type of toxoid that is given to you. So it helps in sustaining an immune response against such a bacteria so that it can clear it off from the system. Um, yeah. Then comes the subunit type of vaccine, which is also an inactivated exotoxin and is used for hepatitis B, pertussis, which causes whooping cough. Bordetella pertussis is what causes it. And you have streptococcal pneumonia as well. So in this case, the specific antigens, they lower the chance of adverse reactions, but the problem is that subunit vaccines are very difficult to develop. So while it's more specific in nature because you are developing a vaccine for only one region, one subunit, which will be good enough to induce the immune response or elicit the immune response, but will be physiologically safe for the individual who is being vaccinated. But their commercial production is quite difficult to do, and therefore it's a challenge that remains open till date. The next subtype is of conjugate vaccines, which is example of which is the Haemophilus influenzae type B, streptococcal pneumoniae. Haemophilus influenzae type B is often known as HIB, HIB, and it helps in priming the infant immune system to recognize other bacteria, basically. We'll be briefly discussing conjugate vaccines also eventually. Other types of vaccines include DNA vaccines. You will find examples of these, but till date, not a lot has been commercially approved. There are a couple of vaccines, particularly for neoplastic diseases, which are very promising, which are potentially going to be implemented for the public soon. But most of them are in clinical testing stages. What they effectively do is that they induce a strong humoral and cellular immune response. They're quite inexpensive manufacturing wise, but yes, they are a little difficult to actually implement for the general public. You also have recombinant vector vaccines, whereby it helps in mimicking the natural infection, resulting in a very strong immune response. So these are the different subtypes. I will not be discussing DNA and recombinant vector vaccines today. You can look these up. They are easy to access online. You'll find a lot of interesting information regarding DNA vaccines, particularly. So if it is something which is quite interesting to you, something you might want to pursue in the future, please go ahead and look at these topics. They're very, very interesting. But yes, they're difficult to comprehensively explain in just one session. So you can look at that. Now we'll come to the topic of immunization. Just give me a minute. Okay, so we are talking about immunization now. So what exactly did we mean by immunization? You saw that terminology. It means the process of eliciting an immune response against a certain pathogen which causes a disease without actually introducing the entire pathogen. You're introducing some variant of it. What does it do? The process of immunization, what does it do? It induces immunity, right? So just a second. Yeah. So if you look at the types, there are two types of immunity which can be induced as a result of vaccination or immunization process. Either it can be passive or it can be active immunity. When we talk about passive immunity, its acquisition can be either natural in nature, whereby you can see that it is from the natural maternal antibodies through the colostrum, where it can be taken up by where an infant takes up the mother's antibodies by their during the process of lactation. And also, of course, it can be from natural previous infection, but that will induce active immunity. When we talk about artificial methods, it can either be by immunoglobulin, or it can be by an antitoxin which we are introducing into the system, or by a humanized monoclonal antibody. Active immunity, apart from a previous infection, can be induced by vaccines. Among vaccines, again, we have attenuated organisms, inactivated organisms, purified microbial macromolecules, cloned microbial antigens expressed as recombinant proteins, or cloned DNA alone or in viral vectors, multivalent complexes, and of course, toxoids. You can find some references being mentioned in this case as well, that apart from the maternal antibodies, you can also get antibody containing solution from human blood obtained by cold ethanol fractionation of large pools of plasma available in intramuscular and intravenous preparations. Then the antibody from antitoxins, these are antibodies derived from serum of animals that have been stimulated with certain specific antigens. Vaccines are a suspension of attenuated live or killed microbes or antigenic portions of them that are presented to a potential host to induce immunity and prevent the disease in the long term. Toxoid is a bacterial toxin that has been modified to become non-toxic, but it still retains the capacity to stimulate the antitoxin formation. So among these processes, how can you actually classify them? So of course, active and passive immunization are two classifications, but apart from that, you can have natural active immunization or artificial active, there is a spelling error, but yeah, artificial active immunization. So first, when we talk about natural active immunization, it is achieved by natural infection with a microorganism. The immune system will eventually create antibodies against the microbe. The next time when there is an infection with the same microbe or same pathogen, then the immune response will be much heightened. It will be quicker, that is, it will be more rapid and it will be more heightened in nature. The example to this is that it's a case in many childhood infections where a person has contracted a certain disease more than once in their lifetime and then they become immune to it in the subsequent um, during subsequent infections. During artificial active immunization, however, it is achieved by vaccination. A vaccine is a biological preparation that provides active acquired immunity to a particular disease. And the intentional administration of this vaccine cocktail is called as vaccination. It has played an important role in the reduction of diseases, especially among children. Here you can see some data which actually represents this. It is the data for measles, which you also might have heard very commonly. It happens, it is not as prevalent as it used to be, say, 50 years ago. So you can, ask your parents or your grandparents at home, and that will give you some perspective about how prevalent measles used to be back in the day. So you can see how over time, see from the 1950s, the number of cases in thousands has been plotted along the y-axis. And you can see how its frequency has initially was quite high and kept on increasing till the um, late 50s, early 60s. And then somewhere in the middle 60s, the vaccine was licensed. And from there on, administration started. And within like 3-4 years, you can see how there has been a steep decline and it virtually came down to a very minimal number by the 80s. And after the 80s, particularly in the 80s, if you see, the number of cases had declined very significantly. So very occasional outbreaks of measles occurred mainly among unvaccinated young children from the 1980s onwards, and among some college students who had only been vaccinated once. They did not get the booster shots for the disease, and only in those cases you could find reports. So overall, what you can see is that an insane and unbelievable decrease had happened in the incidence of a disease just by virtue of this beautiful technology that came in and that identified the potential of vaccination, and people who were willing to take the necessary vaccinations, and how it has dramatically decreased over a period of just a few years, a few decades. Here, what you can see is a sort of chart which shows you the necessary vaccinations as recommended by the Indian Academy of Pediatrics, which I was just mentioning a while back. So this actually has been taken from the IAP or Indian Academy of Pediatrics official website, and they have an advisory committee on vaccines and immunization practices, and this shows you the recommended schedule as of 2020-2021 for children from 0 to 18 years of age. So what you can see is that it has also been color coded. Again, you don't need to memorize this. It's just for your understanding and to relate what you're studying with practical utility, because in the long run, if you want to go into research, that's the goal. So yeah, so that's what you see, that from the from birth till the age of a few weeks, from 6 weeks for 10 weeks, 14 weeks, and so on and so forth, you can see the recommended age for any vaccine and what is the overall catch-up range within which you should definitely take the vaccine, and some special situations whereby certain other vaccines are usually recommended. The most common one is the BCG vaccine, which should be taken right at birth. I'll be coming to the BCG vaccine very soon. Then comes hepatitis B vaccine and all the different doses that are recommended for the same. You have the OPV or oral polio vaccine next. You have DTaP, which is diphtheria, Bordetella pertussis inactivated toxin. The different doses of that, Haemophilus influenzae B, rotavirus influenza, hepatitis A, HPV, cholera, yellow fever, rabies, etc. However, these vaccines, as you can see, this can be taken in special situations, particularly yellow fever vaccine, for example. This is not normally taken up by most individuals, but in some cases they are compulsory. For example, if you are traveling to a country where it is very prevalent, then it is advised and many a times even necessary for you to have those vaccines before you can enter that country, and you need to have the documentation to actually support that. So those are very case specific incidents that I'm talking about. Next, coming to the idea of booster doses, we had just talked about it. It simply put, it means a condition where you have repeated inoculations with a certain immunogen so as to elicit an immune response for that. The most common example of this booster dosage is for the polio vaccine, whereby multiple immunizations or boosters, at least 3 are recommended for the polio vaccine, and they are necessary to ensure that you have an adequate immune response elicited to each of the 3 strains of poliovirus that make up the vaccine. So what you see on the left is a slightly older sort of poster, which was designed for polio, that we are zeroing in on it, one viral strain at a time. For example, the type 2 strain has been eradicated for a very, very long period of time. It was last seen in 1999 in October, and there have been no reports for it since then. So it has now been declared eradicated by the WHO or the World Health Organization. Also, these are some useful terminologies for you to know. Like, of course, the WHO is something we have all familiarized ourselves with after the COVID pandemic. But apart from that, CDC, or the Center for Disease Control, is an Indian body that it's also a governing body that actually regulates this. You have the Indian Academy of Pediatrics for the vaccination schedule for children. So these are all the different governing bodies who are concerned with the implementation of the vaccines that get discovered by other research labs, for example. But yeah, apart from this, you also have other types or other strains or serotypes of this particular virus. So you have type 3, which was last reported in 2012, but it has yet not been completely declared as eradicated, or I think I need to look into this once more because I'm not exactly sure. It has not been reported, but if there's even a single incident, a single case which gets reported for this, then that will be considered as the new date whereby the last scene has to be changed accordingly. But yes, what you do see is the type 1 strain, which is still present. But what I've also put as a supporting data for the fact that polio is going to be the next big thing that gets removed after smallpox. You can see this is the most recent data. I looked it up today, so it's as updated as it can be. But yeah, you can refer to these websites for your own understanding. You can just search end polio, or you can go directly to the Global Polio Eradication Initiative website and you can search there. And you can see how there have been recent cases that have been reported as recent as September 2024 in Pakistan and in Afghanistan. You can see in August, some cases have been reported. So in 2024, a number of cases have been there, approximately 28 cases of wild virus type 1. So there are still a few reports that are definitely there, and from other sources also you can see some cases that have been reported. But certainly you can expect that over a period of time these will decline. And in a few other countries you can see very few reports that have been there in 21 or 22. The last case has been identified, and we can expect this to be gone in the next, let's hope, 10 years. And then we can wait on it for another disease to be completely cleared off just by vaccinations and by people being very cautious about it. So that was what I wanted to highlight. Now, coming back to the idea of whole organism vaccines, basically the types of vaccines, but among them, the first subtype is, of course, for whole organism vaccines. Among these, you have live attenuated avirulent, bacterial, and viral vaccines. So, as the name suggests, these are vaccines against viruses and bacteria, which are disease causing or pathogenic in nature. And because they are live attenuated, it means that they have somehow been altered so that they no longer are able to cause disease. So microorganisms can be attenuated so that they lose their ability to cause significant disease. That is, they lose their ability to be pathogenic in nature, or they lose their pathogenicity. However, they do retain their capacity for transient growth within an inoculated host. It means that if a particular individual is getting inoculated, or if this particular virus is being introduced into the system of a particular individual, then they will be able to grow there, but will not be able to cause the disease that they're usually associated with. So you have disease or pathogen and the type of vaccine being enlisted here. Among bacterial diseases, we have anthrax, cholera, pertussis, plague, tuberculosis, and typhoid. Majorly, you can see that they are inactivated vaccines, but for tuberculosis and typhoid, you have live attenuated vaccines. We will be looking at BCG very soon to understand it in a little more detail, and it's actually quite interesting. And apart from that, for viral diseases, you have hepatitis A, influenza, polio Salk vaccine, rabies, and the rubella virus, against which inactivated virus is given as the type of vaccine in those patients in such cases. And for measles, mumps, polio Sabin vaccine, rotavirus, varicella zoster, or chickenpox, and the yellow fever, you have live attenuated virus, which is produced. Again, please keep in mind the difference between polio Sabin and polio Salk. For Salk, you have inactivated vaccine, whereas for Sabin, you have the live attenuated vaccine. Now let us first understand the different methods of attenuation. When we say attenuation, it means something which is not completely lost, which means that you're not completely killing something off, but you are reducing its potency. There are two methods of attenuation. You have natural and artificial methods of attenuation. Natural attenuation might be due to the inability of a pathogen to cause disease in a given host, but they can immunize a host. That means they can raise antigen-specific antibodies. The example for this, the first vaccine used by Edward Jenner, is of this type. Like you remember, we had just discussed, vaccinia virus inoculation of humans confers immunity to smallpox, which is related to cowpox, but it does not cause cowpox in humans. So we saw how in that case, individuals who were suffering from the cowpox disease, from their pustule, when the fluid that is present within their pustule was introduced into a healthy individual, then such an individual will not suffer from smallpox. So there's a prophylactic action or a protective action of that virus when introduced in another individual. So that is a form of natural attenuation, whereby as humans, we are not interfering in any way. We are simply taking the protective ability of one virus against another one. The second method of attenuation is for artificial cases. So attenuation often can be achieved by growing a pathogenic bacteria or virus for very prolonged periods under unusual or adverse culture conditions. What does it done? So attenuation has multiple mechanisms. What you actually probably came across in the video lectures was that you go for chemical or heat-based inactivation, which is done. So you have mechanical or chemical modes of attenuating it. However, another way, which is what is very relevant for the BCG vaccine, is that you are growing the pathogen under some very unusual condition or very adverse or abnormal condition. So what does it do? When you are changing the condition in which a particular media, in which a particular pathogen is normally able to grow, what you are doing is you are allowing that pathogen to modify itself or transform itself or mutate itself. So this procedure, when you are culturing it under that changed condition for a prolonged period of time, helps in the selection of mutants. These mutants are better suited to grow in those unusual conditions, and therefore they are less capable of growth in the natural host. For example, production of the BCG vaccine or the Bacillus calmette-guerin vaccine. What is it? An attenuated strain of Mycobacterium bovis is called Bacillus calmette-guerin. Again, what you need to keep in mind is that you are not using the Mycobacterium tuberculosis, the pathogenic strain of the virus, of the bacterium. Instead, you are using a different strain which has the immunogenic capacity but is not disease causing in this case. So the attenuated strain was actually developed by growing Mycobacterium bovis on a medium containing increasing concentrations of bile for a period of approximately 13 years. The result of this is that it led to some mutations within the bacteria, as a result of which it was developing into this different variant or this mutant condition, which was called as BCG or Bacillus calmette-guerin. After 13 years, this strain had the capacity to adapt to the growth conditions in strong bile, and now it was sufficiently attenuated and was suitable to be used as a vaccine for tuberculosis. And it could permanently only grow in strong bile. Very transiently, it could grow in the human body. So this is what the BCG vaccine vials actually look like when they are when they were commercially allowed for purchase and for utility. And this on the right, you can see that after the vaccine had been administered, what it looked like. So this is one example of attenuating a pathogenic strain of a particular pathogen for its utility as an immunizer or an immunogen. The other example, which obviously you have come across if you've gone through the lectures, is the use of the polio virus, which is used in, just a second. Yeah. So it is the polio virus, which is used in the Sabin vaccine. That is the attenuated form of the vaccine. And that for that condition, what is done is that growth is facilitated on monkey kidney epithelial cells. So under normal conditions, they should grow on human cells. But in this case, to attenuate or to curb its potential, they are grown on monkey kidney epithelial cells. So that's the way of attenuating it. Following this, we have on the right, you see an image of Albert Sabin, who was the discoverer of this vaccine. And he was at the University of Cincinnati, whereby he's feeding oral polio vaccine or OPV to a child in the early 1960s. And yeah, that is the most commonly used and the most commercially, I would say, relevant form of vaccine that is being extensively implemented in countries across the world, including third world countries such as ours, in India, whereby you can see that you will find a lot of commercial push to, by celebrities and a lot of announcements that are carried out across cities, towns, villages, everywhere to make sure that your child has the vaccine. It's just, 2 drops of that particular vaccine. It's marked on the child's finger to make sure that the vaccine has been administered, and from schools also participate in this to make sure that administration can be done. And it has been probably the most commercially successful vaccine administration across the world. So here you have a side-by-side comparison of the advantages and disadvantages of live attenuated vaccines. So while these vaccines, they provide a very prolonged immune system exposure to the individual epitopes on attenuated organisms, that helps in increasing the immunogenicity and stimulating the memory aspect of the immune system, and thereby overall producing a very strong immune response to a particular pathogen. As a consequence, only single immunization is often very useful in these cases, and it eliminates, in most cases, a need for an additional repeated booster. One exception to this is the Sabin polio vaccine, whereby it is recommended that at least 3 doses, if not more, is suggested. Um, but yeah, most cases you do not need repeated boosters for live attenuated vaccines. And this property is a major advantage, especially in third world countries where epidemiological studies show that roughly 20% of individuals do not actually turn up for subsequent boosters. And so their immunization is actually incomplete and that's certainly not desirable. The ability of many attenuated vaccines to replicate within host cells makes them particularly suitable for inducing a cell-mediated response. So that's of course, the sustenance of such a response will be much long-term, and therefore is very, very desirable, of course. Following that, we have some comparative disadvantages as well. The possibility that they can revert to their virulent form certainly remains a challenge. It's something you cannot always predict. It's all obviously remains a possibility, and that's a risk that people obviously take. The rate of reversion of the Sabin polio vaccine, which is the oral polio vaccine, the attenuated form, leading to subsequent paralytic polio, is about 1 case in 2.4 million doses of the vaccine. So while that might seem like like a very affordable statistic, so to say, it is a significant number, because if you consider the world's population at large, um, it is a statistically significant number. And if there is even a single case where a particular person gets infected with the same, then the approval for such vaccines goes down. This is a major hindrance to the complete eradication of paralytic polio through the use of OPV, because that chance, no matter how small, still persists. And of course, if it is, somebody you would not be very accepting of such a vaccine, and therefore a lot of people often hesitate before taking the vaccine as well. But certainly that's a challenge. Everything comes with a challenge. It's not foolproof. This reversion implies that the pathogenic forms of the virus are being passed by a few immunized individuals, and they can find their way into the water supply at large, and they can potentially allow the poliovirus to cause infection. This possibility has led to the exclusive use of the inactivated polio vaccine in many first world countries. But certainly that's a difficult thing to commercialize, because that's expensive, it's difficult to administer, and the oral polio vaccine was a lot easier to actually implement within a large population. So while it has its disadvantages, it is most commonly used, especially in populations that are quite large in countries like ours in India. But yes, countries that can afford it, they prefer going for the inactivated, the Salk polio vaccine. And yes, if that can be made, if that can be implemented worldwide, that would be a wonderful achievement. Apart from that, the second subtype is of inactivated or killed vaccines. So, of course, inactivation of the pathogen has to be done by some means. So, it is usually by heat or by chemical means, so that it induces an immune response but is no longer capable of replicating in the host. It is critically important to maintain the structure of the epitopes, because that is what will induce the immune response upon being administered into an individual. So it's important to maintain the structure of these surface antigen epitopes during this inactivation process. This was a key consideration to be made, a serious complication with the first Salk vaccines that arose when formaldehyde or HCHO, which is a chemical means of inactivating the pathogen, failed to kill all the virus in two vaccine batches or vaccine lots, which caused paralytic polio in a very high percentage of recipients. Again, this is something you can consider as a technical fallacy, and it cannot always be um, as much as possible, as much as the regulations are kept in check, it's always a possibility that something goes wrong. That's what had initially happened. So that led to more strict regulations and more strict maintenance and, you know, checking before administration is being actually done. So here you can see a comparison between attenuated and inactivated vaccines and the characteristic with respect to certain characteristic features. The first being production. So selection for avirulent organisms. Virulent pathogen is grown under adverse culture conditions or prolonged passage of a virulent human pathogen through different hosts, like you saw for the polio vaccine. In comparison for inactivated vaccines, the virulent pathogen is inactivated either by heat or chemicals or irradiating with gamma rays, so that it is now altered or mutated and can no longer cause disease. Second is booster requirement. Usually attenuated vaccines generally require only a single booster, but inactivated vaccines require multiple boosters because they cannot persist within an individual system for a very long period of time. Relative stability for inactivated vaccines is a lot more because they're not living in organism, versus attenuated vaccines have a much more hectic maintenance associated with them. Then the type of immunity which gets induced for attenuated vaccines is both humoral as well as cell mediated, because it is still a living organism, so to say. But in comparison, if you look at the inactivated vaccine or the heat killed vaccines, for example, they are majorly the type of immune response in that case is only B cell mediated immunity, otherwise known as the humoral branch of immunity. The reversion tendency in case of attenuated vaccines is a lot higher. They can revert to the virulent form because they have not been completely killed. Whereas in comparison for inactivated vaccines, the possibility is much, much, much lesser. Oops, sorry. The next subtype is of subunit vaccines. So you have purified macromolecules like we talked about previously, toxoids from diphtheria or from tetanus. So diphtheria or tetanus, as the name indicates, they are toxins that are being synthesized and secreted, and they can be inactivated and commercially used. Subunit vaccines are also made for capsular polysaccharides, especially for Haemophilus influenzae type B polysaccharide along with a protein carrier, which is also known as also an example of conjugate vaccines, because the polysaccharide by itself is not immunogenic enough. So you actually tag it along with something which has a higher molecular weight, which has more available epitopes, which will be capable of inducing an immune response in such cases, and or some other sort of protein carrier. Then you have Neisseria meningitidis, which is a polysaccharide by itself. Streptococcus pneumoniae, 23 distinct capsular polysaccharides are present, which is one of the main reasons why till date influenza has not been completely eradicated, and it is nearly impossible to do so. It seems impossible to do so because not just because of the type of polysaccharides, but because of the nature of the virus by the nature of the organism by itself. But the influenza virus, sorry, I actually mixed those two. Pneumonia is actually difficult to tackle because of all these different polysaccharides. You cannot, uh, while it is potentially very promising that you can just design a vaccine against the polysaccharide, the combinatorial availability of the surface polysaccharides can be different, and therefore it's difficult to target them. What I instead meant about the influenza virus was the fact that the virus has a very strange type of genome. It has a fragmented genome, which can undergo, which can lead to the presentation of different forms of the surface epitopes, and therefore targeting it by any one sort of vaccine is an impossible challenge that we have still not been able to properly figure out. Um, this is a sort of flow chart which shows you the way in which these toxoid vaccines actually can work. So you have bacteria which are secreting exotoxins. You can purify them, these bacterial exotoxins, and then use some sort of agent to inactivate or kill them. So majorly it is by heat or by use of chemical reagents like formaldehyde, which turns them sort of inactive or avirulent form of the toxin to form a toxoid. What is the condition that you actually want, or what is desirable, is that you want to achieve detoxification without excessively modifying the epitope structure. The most common examples being diphtheria and tetanus toxoids. Finally, I think, yeah, that's the last slide for today. We have conjugate vaccines. So a conjugate vaccine is a type of subunit vaccine which combines a weak antigen with a strong antigen as a carrier so that the immune system has a stronger response to the weak one. The example for this is the vaccine for Haemophilus influenzae type B, or the HIB, a major cause of bacterial meningitis, which causes deafness and neurological defects in children who are less than 5 years of age. And what it basically consists of is the HIB capsular polysaccharide covalently linked to a protein carrier, which is the tetanus toxoid. So here you can see the Haemophilus influenzae bacteria. On the surface, you can see these polysaccharide molecules which are present. So that entire polysaccharide is linked to some sort of a protein carrier. Majorly, that is the tetanus toxoid, because that will simultaneously immunize them against the tetanus toxoid as well. Yeah, and it also increases the immunogenicity of the entire immunogen rather than just having the polysaccharide by itself, and therefore that has more potential. So this polysaccharide protein conjugate vaccine is more immunogenic, and therefore they are both administered together, and it's often very useful and is observed to be quite promising, because especially the induction of meningitis, sorry, by Neisseria meningitidis or by Haemophilus influenzae in children is extremely dangerous. You can find it in growing ages as well, or in adults as well, but the deficits that set in very young children are quite extensive. So yeah, I think with this we have reached the last slide. I will share some sample questions, and then we'll end the class for today. The reason classes are ending so early is because we don't have anybody joining in most cases, and I understand that time does remain a constraint for a lot of people. So yeah, I guess that's all right. Just a second. I cannot find that file right now. Just excuse me a minute. Okay, so I'll just go to this week's assignment itself, I'll not be discussing the answers, but I'll try to help you understand how you can answer these questions. So majorly what you need to understand here is during the manufacturing of vaccines, very small amounts of other components such as adjuvants, preservatives, and stabilizers are added along with the antigen to prevent contamination and increase the immune response. So all are commonly used adjuvants in vaccine except which of the following. So if you go through the lectures very properly, you'll be able to address that, that what is the adjuvant that is most commonly used, which of them is quite unusual. You'll find some human approved adjuvants, and you'll also find some which are approved for other organisms as well. I'm trying not to give away the name directly, but yes, you can look at the different types of adjuvants. Apart from that, of course, preservatives are necessary so as to sustain the vaccines over a very long period of time, so that over, say for example, synthesis is, for example, manufacturing is being done in for a particular vaccine is being done at Salk Institute. Now, from there, it to be imported into India. In India, it will reach some major hubs, say, for example, Delhi. From Delhi, it needs to go to a certain vaccine supply unit somewhere in Madhya Pradesh, and then in Madhya Pradesh it has to reach a certain hospital, and then it has to stay there. So you can see the amount of time that the vaccine has to sustain, and its storage should be under the ideal condition so that the protein does not get degraded. Particularly, this is relevant if you are doing some passive administration, because in that case, antibodies are your object of concern. They are what will induce that immunity, and therefore the maintenance of antibodies is very crucial. Even in the lab conditions, if you are familiar with it, when we work with antibodies in the lab, we make sure that they are constantly being maintained at a cold temperature, majorly because they will degrade otherwise, their proteins. So we maintain them at least 4 degrees, and storage in some cases is at -20. Recommended storage is at -20, so that they remain in a frozen state and they do not undergo degradation. So yeah, you need preservatives and of course stabilizers also. Sorry, stabilizers also for the same purpose. Then we have been asked to choose the correct statement about the Sabin polio vaccine. It consists of 3 attenuated strains of poliovirus, 3 inactivated strains of poliovirus. It is administered orally. It induces production of secretory IgA in the intestine. It requires booster doses, or it requires a single immunizing dose. So again, I have repeatedly stressed on the difference and the key distinguishing points between Sabin and Salk vaccine. I hope that was clear enough for you. If not, you can again go back to the video lectures by Professor Shubhi. Or of course, you can look it up online as well to study a little bit more about the discovery, the current implementation, how prevalent these vaccines are, and that should be a good enough strategy for you to answer and understand and answer questions like this. Monoclonal antibodies or MABs are used as therapeutic agents because they can specifically target aberrant cells. But there is a major disadvantage. Monoclonal antibodies raised in mouse are expected to develop an immune response when injected into humans. To prevent this difficulty, monoclonal antibodies are humanized by doing which of the following. So humanization of antibodies is something that Professor Shadeep has already discussed. I did not bring it up in today's session, but again, you can read up a little bit on this just as a hint. You can study about SCFVs or single chain variable fragments, and that should help you understand the basics of it. And of course, your very basic knowledge of antibody structure, which we have extensively been discussing since week 2, I think, should certainly help you understand it. And you should understand what should be the minimal region which is required to induce an immune response and how it can be humanized to make it more effective. Immunotoxins are what? antitoxins, antibody for specific antigens stacked with toxins, formaldehyde inactivator toxin, low immunogenic toxins, or inhibitory chain of bacterial toxins that can induce it. Okay, I cannot address this question directly. You can refer to, I think, the third video for this week, the lectures for this week, if you are not clear about it. But yeah, it's a very direct, factual question. So maybe I'll refrain from discussing it. And we'll take it up very soon in the next week's session. And maybe that should help you out. Maternal antibodies acquired transplacentally during fetal life. Transplacentally means across the placenta during fetal development, and by the intestinal absorption of colostral Igs. Colostral Igs means they are immunoglobulins which have been absorbed from the colostrum of the mother by the infant. They are an example of natural active immunization, natural passive immunization, artificial active immunization, artificial passive immunization. You can refer to the table that I had shown today, or just conceptually it should be quite easy for you to tackle as well, that from the mother's body, antibodies are being absorbed by a fetus, either via the placenta or via consuming the milk from a lactating mother. So yeah, that should answer your question. Then we have toxoid vaccines, which are prepared by inactivating bacterial toxins by treating them with formalin, HCHO. One such example of toxoid vaccine is which of the following? We have discussed this extensively, the two types of toxoid vaccines most commonly implemented. So you should be able to answer that as well. The conjugated vaccine for Hib or Haemophilus influenzae type B, which causes bacterial meningitis in children under 5 years of age, is prepared by linking a type B capsular polysaccharide to another type B capsular polysaccharide, a protein carrier, a lipopolysaccharide carrier, or a carbohydrate carrier. So again, this was there in the last slide, if you refer to it, and that should be very good enough for you to understand. But yes, conceptually, you should remember that as the prime example for conjugate vaccines. So that enhances the immunogenicity of the vaccine at large, and that can be commercially utilized as well. The adjuvant, which is approved by the FDA in the United States for use in marketed human vaccines, is Freund's complete adjuvant, bacterial polysaccharides, alum, or MF59. This was repeatedly discussed by Professor Ghosh in the lectures. But yeah, if you're not familiar with it, you should have known one of the agents very commonly when you study chemistry also, for example, which is compatible with the physiological system of any human being. So that is the most commonly used adjuvant, which does not elicit any non-specific immune responses, is acceptable to the human body, and is efficient at doing its job as an adjuvant. Edward Jenner made the first smallpox vaccine by performing an unethical experiment, like I just mentioned it during today's discussion as well, that you would not be able to do that because now you are answerable to a lot of governing bodies, but certainly he was able to do that back then. The material used by him for inoculation was which of the following? A carrier vaccine, a subunit vaccine, an inactivated virus vaccine, or a live attenuated virus vaccine? Now, that's a great question, because what you need to keep in mind is that this was done in the 1800s. So he did not actually do it with a lot of approval. So you don't have a clear classification for what was done by him. So you need to keep in mind what was the material that was taken, what was the condition that the material was in at that point of time, and what was the way in which it was administered, and you should get your answer. We'll discuss it in the next week, but yes, that's a very interesting question to a fact that we have repeatedly discussed. And finally, the last question, for successful vaccine discovery, viruses are attenuated by which of the following? Passage of live viruses from cell to cell, searching for less virulent mutants, using low temperatures to select for cold adapted mutants, Passage of live viruses from cell to cell, searching for less virulent mutants and using high temperature to select heat adapted mutants, direct random mutagenesis to create a less virulent mutant, formaldehyde, and several alkylating agents. So you will find some similar options, but yes, again, you should go for something which seems the most obvious to you. Again, something we have discussed both in today's session and previously as well. So that should help you arrive at the answer to this question. I'll not say more about this because it's something we have discussed, the way in which viruses have been attenuated and what should be the successful, what should be the ideal strategy for making a successful vaccine. So with this, we come to an end for today's session. So basically what we did today was to look at the way in which vaccines have been used, how they were discovered initially, what has been the strategy for their implementation around the world, what is the currently recommended dosage, what is the frequency at which these need to be taken up, what are the changes that are required because or that have been required over time to make them more efficient at being implemented worldwide. What are the challenges faced with each subtype and how what are the advantages as well of using those subtypes. So all of that has been taken together. What you need to keep in mind is the marvelous contribution that these discoveries have made. The most recent example being the COVID-19 disease and how transformative it was that the administration of vaccines were able to do. What you also need to understand that every discovery comes with its own shortcomings. The shortcomings of the COVID vaccines are something we will learn over time, and we'll have to deal with it because it was a pandemic that was causing havoc around the world for months at end. So curbing it or mitigating the effects of that infection was top priority at that stage, and whatever comes along with it is a challenge we take as a society at large. So with this, I would like to end today's session and officially the last live interaction session for this course. So thank you so much for joining either here in the live interaction sessions or joining via YouTube or just going through my slides for your own studies, whatever is more convenient to you. The job is to help out students however we can. And I hope it was a good experience learning for you and it was helpful. And I hope I was able to add to what was already taught by the professors and the sessions were at least to some extent useful for you. So thank you so much. Thank you for being along this journey. We'll meet next week, and we'll conclude our sessions for this entire course. Thank you so much. Have a good day.